So this topic is about the biosynthesis of alkaloid, which we will discuss the different derivatives and the functions involved for their biosynthesis. Firstly, the alkaloid is a nitrogenous organic molecule that has a pharmacological effect on human and animals. They are a class compound which typically contain nitrogen and have a complex ring structure. Alkaloids are found as secondary metabolites in the plant, for example Venca and Datura, in animals, for example in shellfish, in fungi, and can be extracted from their sources by treatment with acids, usually hydrochloric acids and sulfuric acids through organic acids such as the malic acid and the citric acids. The first is the ornithine derivatives. In plant, it is formed mainly from the L-glutamate. Ornithine supply as a C and N building block to the alkaloid, principally as a five-member pyrrolidine ring system, but also as a part of the tropine alkaloid. The derivatives such as in the tobacco plants and in the solutionous family of the plant cells. So this is the general diagram of this alkaloid. The second is the biosynthesis of the tropane. The starting compound of this synthesis is the ornithine, and methyl-ornithine is the first intermediate. Ornithine converts into tropine, and then phenylalanine is converted into the tropic acid. Now the tropine react with the tropic acid and it will produce the hexenamide or the atropine. So this is the general diagram or the general explanation of the tropine and the tropic acid which react to produce the alkaloid derivative. Other derivative is the lysine derivatives. L-lysine is the homologous of the L-ornithine, and it to function as an alkaloid precursor using pathway analogous to those noted for the ornithine. Lysine supply a CN building block to the alkaloid, principally as a six-member piperidine ring system. The derivatives such as the lupine, lupinine, and anabasine pelletrines alkaloids. Again, the lycopodium are substances obtained from the lycopodium species example. So this is the lysine derivatives. Phenylalanine derivatives. L-phenylalanine is less frequently used and usually it contribute only in carbon atom units without providing a nitrogen atom from its amino group. Here the example is the epidrine, the main alkaloids species of the Epidera. Epidrininia is a prime example of the phenylalanine derivatives. This is a general diagram of the phenylalanine derivative. Tyrosine derivatives. The first essential intermediate is dopamine. Dopamine is the precursor in the biosynthesis of the pepramine, herbamine, and the morphine. L-phenylalanine is also the precursor of the formation of the epidyrene. Biosynthesis of the morphine. A ring of similar compound like the opium alkaloid, the thymine, codeine, etc. are derivative during the formation of morphine. Tyrosine is the precursor of the porphine, a potent analogous. Tyrosine is the precursor of codeine, a anti-tissues. Biosynthesis of tryptophan derivatives. The tryptophan derivatives correspond to 25% of all non-alkaloids and many of them are medicinally valuable. Tryptophan and its decarboxylate products like the tryptamine are precursor for the biosynthesis of broad range of indole alkaloid of which the Venca and the Rufilum alkaloids are example and also is the alkaloid belonging to the family like the Epicynacea, Loganaceae and the Rubiaceae. This is the general example of the tryptophan. Biosynthesis covalent alkaloids. Some of the most remarkable examples of tryptophanate indole alkaloid modifications are to be found in the genus Sencola rubiaceus is the alkaloid quinidine, cancertodine, and for the anti-malarial properties. Biosynthesis of lysergic acid derivative. The building block for lysergic acid are tryptophan less than the carboxylic group and the isoprene unit. Alkylation of tryptophan with dimethyl diphosphate give 4-dimethyltryptophan, which then undergo N-methylation. Formation of the tetracyclic ring system of lysogenic acid is known to proceed through clincoline 1 and agroclivine. So this is a general example of the lysergic acid derivatives.